Hello everyone and welcome back. In this video, we'll be learning to create a BioPython application and few other terms that are required for us to know before getting with further course of the video. So let's get started. The first topic of the day is parsing. Data parsing refers to identifying tokens within a data instance and searching for patterns. A parser segregates each word, determines the word's relationship to previously defined token sets, and forms patterns based on sequence of tokens. In bioinformatics, dealing with biological data format is large part of the work. Trying to parse these files can also be frustrating because formats can change quite frequently. Formats may also contain subtleties that makes them incompatible to work with. In BioPython, bio.seek.io provides a wide range of uniform interfaces for importing and exporting data. In this format, seek.record objects are the only format that can handle sequences. Lowercase strings are used while specifying the format. The same formats are also supported by bio.align.io, about which we'll be discussing in the upcoming videos. Examples of few formats are FASTA, GB, Swiss, and PHT like we saw in the previous video. There are also other commonly used extensions such as PDB for atom of protein structural determination, FastQ, a variant of FASTA with Sanger, and FastQ Illumina used for storing Phred scores. Those were all the concepts and necessary terms needed for us to know before getting into creating a BioPython application. Now we will create a simple BioPython application to parse a bioinformatics file and print its content. So this helps towards understanding the general concept of BioPython and its usage in bioinformatics. So I have my Google Colab page open here and I have named my file as creating a BioPython application. So I have my bio package installed already. You need to do this step every time before you start with a function or a program. So let's run the bio package and get it downloaded. It says that requirement is already satisfied. So our bio package is all set to be used for further functions. So right after getting my bio package downloaded, our next step will be to create a sample sequence file. And I've already created something here called learnbio.fasta. So I obtained the sequence from an open source website and I named it learnbio, and FASTA is the extension or the file format of the sequence I created. So here is the sequence. And FASTA originates from a bioinformatics software company name, and hence it is named so. It has multiple sequence that is arranged one by one with a unique ID, name, description, and actual sequence information. So right after creating this file, we are going to upload it in our Google Colab node. So how do we upload it? You have 3 lines over here, click it. You have something called files over here. And I have something called sample_data, a folder. You may have whatever folder you want in your Google Colab notebook. And I've uploaded my learnbio.fasta file inside the sample data. How do we upload it? You're going to right click the sample_data and upload. You will have to copy the path of learnbio.fasta, whatever file that you're going to upload, the sequence file, and you'll have to save it for your future purposes when you're going to retrieve it by opening in your program. So step 2 will be is to copy the following code into a new cell in the notebook. And don't worry about the code. We'll now be digging deeper into the program to see what each line is all about. So line 1. Line 1 imports the parse class from bio.seek.io module. Reading and writing sequence files in different formats are supported by bio.seek.io module, and the sequence files are parsed using parse class. So line 2. In the line 2, seek record class available in the bio seek record module is imported for manipulating the sequence, and seek record represents a particular sequence from sequence file. So that is what is line 2 is all about. And in line 3, it's just importing the seek class available in the bioseek module. So line 5. So here is the pathway that we have copied before of the sequence file that we have uploaded in the Google Colab notebook from our local source. So we are just opening the sequence file that we have uploaded by using regular Python function open. So line 7 is just about parsing the content of sequence and returning it as a list of seek record object. The contents is returned as seek record objects. So from lines 8 to 13, it is just loops over the record and prints the attributes of the sequence like ID name, description, sequence data, etc. So now we'll run the program and see the output it gives us. Like mentioned above, the following attributes have been printed in the output like ID, name, description, annotation, sequence data. So the next video will be discussing in deeper about sequences from biology and how to incorporate them into BioPython. Thank you.